talk about sort of the basic definitions we get when we start talking about both neural network structures and various pieces there, as well as asking questions of how does this look like in terms of basic neuroscience. And so I just want to sort of go over these pieces and sort of go over the connections in a set of terms that are pretty helpful here. Now, many people get used to seeing basics of neural networks, and you'll have a whole input, and usually the approach you look at this is you say, I have a neuron, and that neuron element will take a set of inputs that are weighted, and then I sum them together, and that's what I get in terms of a particular output. And I get a particular output Y. And this neuron is basically meant to be some sort of, you might say, activation function, some sort of nonlinear function, usually going basically linear point to point in this case. So this would be sort of scalar. Now it can also be a layer if I have a whole vector of y's on the output, but it's still going to be point to point. So if it was a more vector outputs, it's going to be sort of point to point outputs. And so you kind of talk about that as a layer. And so you'd say, well, that's what my neuron looks like. And then I have a set of constant weighting values, which then weight sort of these inputs as they're getting summed together. Now these weights are very important because these are the things that we train or learn in sort of learning algorithms. So when you talk about neural network learning or machine learning or all of the other varieties, this is what you're going to be changing. And you're going to be saying, well, what kind of algorithm can I build to do that? Now it's interesting because this will be called a neuron and this will be called a synapse, maybe the summations part of the neuron. Interesting questions, and it's usually a bit fuzzy when people look at this. But if you actually go into the neuroscience, you ask, what would I see there? Well, I would in fact see something that looks like a neuron, and in fact it would usually have sort of a spot that would be sort of the biggest point of it. Usually it's called the soma. Interestingly enough, you'd almost think that means body, because soma comes from the Greek word soma, to mean body. And so that's sort of the center of it. And that will have a single output, and that goes out on an axon. Now you'll talk about it either being spike or spikes or an event, and as a single output, but it's still part of the cell. It's still going along. That structure, if it's going a long distance, may have some additional active components to basically repeat that event to make sure that it keeps its fidelity. But otherwise, this is basically where you're going to start. All right, so on the other side of it, you'll also have what is called the dendrite. Again, also coming from the Greek word dendros, meaning tree. And so it looks like often like there'll be trees or this whole sort of arborization of stuff, and there's whole different structures and all different neurons and different parts of, say, even human cortex, or even in the non-cortical structures, you see all sorts of interesting things there. And the interesting part is when you say, well, how do I get the inputs from another synapse? Well, that's going to come in through, there'll be other axons coming from other neurons, and even potentially the same neuron. This certainly will happen. And there'll be different kinds of regions called synapses, which will be small junctions between the cells, between the two, such that I can have chemicals go across it so I can get signal transduction. And that will get me sort of the communication, and in each of these cases I will be able to have change and modification over long term. It's not quite a multiplication though. It is a multiplication of a sort, but there's a lot more operations going on in each of these things. And so it's very interesting when you start to say, well, this equals to this. You really have to think hard about, well, okay, what approximations am I making? How much of it and how little of it am I doing? And yet, this is where it has very old roots going back to McCullough and Pitts in 1943, where this was their first attempt to describe a neuron because that was what they had. But at the same time, there's a lot more understood over many, many years now, many, many decades. And so lots of different questions come up, and this makes it so much fun to kind of look at all of these questions and all these things in between.